Chromatography is a technique for separating mixture components based on their movement through a stationary phase using a liquid or gas mobile phase. It is widely used to separate, identify, and analyze chemical components in complex mixtures. Principles of chromatography include: mixtures contain multiple components. Unknown compounds must be separated for identification. Components vary in their intermolecular attractions, and some are attracted to the stationary phase, while others are more attracted to the mobile phase, leading to separation. In the pharmaceutical industry, chromatography is essential for analyzing and purifying compounds. The process involves sample preparation, dissolving the compound in a suitable solvent. Second, technique selection, choosing methods like HPLC, GC, or TLC based on compound properties. Third, phase selection, selecting stationary and mobile phases for optimal separation. Fourth, setup column, baking the stationary phase in a column or applying it to a plate, then injecting the sample. Fifth is separation. Components separate based on their affinity for the stationary or mobile phase. Sixth is detection, by using UV, fluorescence, or mass spectrometry to identify components. Seventh is analysis, evaluating purity, concentration, and identity with optional isolation for purified compounds. 8 is quality control, by ensuring product safety, consistency, and detecting impurities.
For the problem statement, chromatography is a crucial analytical technique in the pharmaceutical industry used to separate and analyze complex mixtures such as drug formulations. In the pharmaceutical industry, chromatography plays a key role in ensuring the quality, safety, and effectiveness of drugs. However, despite its importance, not many people are familiar with the topic of chromatography, not to mention its importance in the industry. This video focuses on the topic of chromatography and the importance of chromatography, especially in pharmaceutical industry, with the goal of making it more widely known.
In the pharmaceutical industry, chromatography is used to ensure the quality and purity of drugs.
Imagine you are spending the day with your friends and have decided to go to the new art museum. You'd like to experience the whole museum with your friends, but some members of the group are more interested in the gift shop, while others are true art aficionados and will take longer to go through the museum. The museum has a predetermined path through multiple rooms, each of which has interesting works of art. So, you all decide to split up and move through the museum at your own pace. After a few minutes, the first group of your friends has made it to the gift shop at the end. Your group spends a little more time appreciating the art and reaches the end a little while later. As the museum is about to close, most of the last group hasn't even made it halfway through the full experience. In fact, some have even become trapped in the artwork. This is exactly how chromatography works. With the art museum being the column, you and your friends being the compounds undergoing separation, your interest in the art relating to the stationary phase interaction, and everyone's desire to get to the gift shop afterward equating the mobile phase. In solid liquid chromatography, such as flash or HPLC, molecules can bind to the stationary phase. This is known as irreversible adsorption, meaning your friend is stuck there until they replace the artwork. Truly an inconvenience and an expensive one at that. In our museum analogy, that means demolishing the museum, constructing a new one, filling it with new art, and letting people in, knowing that each time you'll have to repeat the process again. With CPC, this problem is much easier to address. Imagine the same art museum, but in order to remove those art aficionados, each night the museum cleans up by flushing out everything that's left inside. Art with new pieces for the next day's patrons.
For example, in a liquid chromatography (LC) test, we can analyze a drug's active pharmaceutical ingredient (API) and check for impurities or degradation products. Let's take a closer look at how this works. Imagine you have a drug formulation. The chromatogram will show a large peak corresponding to the API, which is the main active substance of the drug. Smaller peaks represent any impurities or byproducts present in the formulation. By examining the size of these peaks, scientists can quantify the amount of each component in the drug. If an impurity peak is too large or exceeds regulatory limits, it signals a problem. It could indicate that the drug is contaminated, degraded, or improperly manufactured. This data is crucial for manufacturers as it allows them to adjust production processes, remove contaminants, or improve the formulation before releasing the drug to the market. In fact, chromatography ensures that drugs meet strictly regulatory standards, guaranteeing their safety and effectiveness for patients. In summary, chromatography is an essential tool in the pharmaceutical industry. It separates, identifies, and quantifies the components of drug formulations, ensuring that each product is of the highest quality, safe for use, and meets the required standards.
So, as the conclusion, chromatography in drug development has played an especially important role in pharmaceuticals, serving as a cornerstone for understanding and refining the composition of compounds, contributing to advancements beyond the scope of drug development. As technology advances, chromatography continues to evolve, offering innovative solutions to meet the industry's growing demands for efficiency and reliability. Thanks to the high sensitivity and accuracy, multiple chromatographic detection methods have been widely used in the analysis of pharmaceuticals.